Hi, and thanks for joining for my presentation. My name is Maddie Ostwald, and I'm a PhD candidate in the Fewell lab at Arizona State University. Today, I'm going to be talking about relatedness in carpenter bee societies, and in particular, how dispersal patterns can reduce relatedness within groups. A central question in the study of animal behavior asks why animals form social groups, despite the intrinsic costs of resource sharing. For animals that live in family groups, inclusive fitness benefits of helping relatives can explain the evolution of group living. However, many animals form groups with distant relatives or even non-relatives. These systems, which evolve in the absence of strong inclusive fitness benefits, raise important questions about the evolution of social behavior. These systems also have the potential to illuminate alternative evolutionary paths to group living, especially those that are driven by ecological factors. The carpenter bees represent a really promising system for addressing these questions about the evolutionary origins of sociality. While some carpenter bees are solitary, many form small nesting groups characterized by dominance interactions and high reproductive skew. These groups are interesting and unique because non-reproductive subordinates don't really act like typical social insect workers. They offer little to no help with provisioning and nest construction, and instead may be waiting for opportunities to inherit the nest or assume dominance. Due to intense competition over access to nests, females may disperse from their natal nests and join neighboring nests of non-relatives, perhaps to usurp the resident dominant or advance their position in the reproductive queue. This dispersal behavior could potentially dilute relatedness in nesting groups. Vickreck and Richards found that the eastern carpenter bee may actually avoid nesting with relatives to reduce costs of competing with kin. I investigated the social and genetic structure of the facultatively social carpenter bee, Xylocopa sonorina. So in this study, we asked how dynamic group membership arising from dispersal behavior shapes relatedness within groups of the valley carpenter bee. We addressed this question using complementary behavioral and genetics approaches. We used mark recapture techniques to observe the dispersal and nest switching behavior that may dilute within group relatedness. Then we assessed genetic relatedness itself using microsatellite analysis. So first I'll talk about the behavioral observations. So we chose a single focal log, which contained about 40 active nests during the reproductive season of 2021. We caught female bees at nest entrances and gave them each a unique paint marking, then observed and recaptured them throughout the reproductive season, starting when activity resumed following winter quiescence and ending just before the next generation of offspring emerged. Each time a bee was observed, we recorded her identity and the ID of the nest that she was either entering or exiting from. So in total, we marked 75 unique female bees, and we estimated that there are around 150 females in this aggregation, so about half of all bees were marked. And we observed 47 of these 75 females on more than one occasion, and the rest were not recaptured after initial marking. So of these 47 bees, 31 or 2/3 were observed at multiple nests. This suggests that the majority of bees disperse or switch nests at some point during the reproductive season. Many of these were observed at 2 different nests, but some occupied as many as 4 or 5 nests between mid-March and early May. And it's important to note that this data represents only a snapshot of the activity of these bees, totaling about 30 hours of observation during times of peak foraging activity spread across several weeks in the spring. Because of this, we expect that this is a conservative estimate of nest switching behavior. The number of times we observed a particular bee is positively correlated with the number of nests she was observed visiting. This suggests that more intensive observation would reveal even more frequent nest switching, so it's likely that even greater than 66% of females relocate over the course of the reproductive season. Now, having confirmed that group membership is highly dynamic in this species, we can address the question of how this dispersal behavior affects relatedness in nesting groups. To that end, we collected genetic samples from female bees at the beginning of the reproductive season, when we expect the majority of dispersal and nest switching to happen. We sampled over 2 years and used the same focal aggregation that we used for our mark recapture data collection. We extracted DNA from these samples and amplified them at 6 microsatellite regions that had been previously characterized in the congener Xylocopa virginica. We then calculated relative relatedness according to the Queller and Goodnight method. It's important to note that relative relatedness is not equivalent to the more familiar R value of relatedness, and instead this value compares relatedness as a function of shared loci relative to the average relatedness of the sampled population, and allows us to compare the relative level of relatedness within groups versus between groups. So we found mixed results across sampling years. Each point here represents a pairwise comparison between 2 female bees, either in different nests or from the same nest. So a relative relatedness value of 0 refers to average relatedness between any 2 bees in the sample. Negative values mean that bees are less related than the average relatedness in the sample, and positive values mean they are more related on average than average. So in 2019, we found no difference in relative relatedness between versus within nests. So in other words, bees were no more related to their nestmates than they were to their non-nestmates. In 2020, however, we found that nestmates were significantly more related than non-nestmates, perhaps suggesting not all bees dispersed from their natal nests at the time of sampling. It could be that variation in phenology across years or in population density and the degree of interspecific competition could explain this variation in relatedness across years. However, this data suggests that close relatedness is probably not the major factor driving group formation in the species, if groups with low relatedness can exist and even dominate in some years. So to summarize, we use mark recapture techniques to demonstrate that nest switching is a dominant strategy in this species, with more than half of all bees relocating at some point during the reproductive season. As bees disperse away from their natal nests to join other groups, we expect that within group relatedness will decrease. We found variable patterns of relatedness across sampling years, suggesting that bees may nest either with close relatives or with non-relatives. And future work should examine how relatedness shifts throughout the year, particularly with the aid of genetic data, genetic markers characterized for this species instead of related species. And finally, this work emphasizes an important possible role for ecological factors in shaping sociality in these species. Because females may disperse away from family groups, inclusive fitness benefits may be insufficient to explain the formation of social groups in the valley carpenter bee. Thank you so much for listening, and thank you especially to my co-authors and funding sources. Please feel free to reach out to me with any questions you might have about this study or any of my work on the ecological drivers of group living in non-king insect groups.